Hello everyone, today we would like to introduce our article published in iMetaomics, titled Impact of Dietary Live Microbes and Non-Dietary Prebiotic/Probiotic Intake on Osteoarthritis and Rheumatoid Arthritis Development, Stratified Findings from NHANES Data. This article reveals the role of dietary live microbes and non-dietary prebiotic/probiotic intake in the development of osteoarthritis and rheumatoid arthritis by analyzing data from the NHANES database. Osteoarthritis and rheumatoid arthritis are diseases influencing the joints, their surrounding tissues, and other connective tissues. The etiology of these diseases remains partially unclear, with some research indicating that the gut microbiota may play a key role in the occurrence and development of these diseases. On the other hand, the potential for dietary and therapeutic intervention in OA and RA using the gut microbiota is gaining attention. Nevertheless, more in-depth cross-sectional studies are required to investigate the connection between consuming dietary live microbes and non-dietary prebiotic/probiotic in OA and RA. Therefore, they utilized data from the NHANES to carry out a population-based cross-sectional analysis. The objective of the research was to clarify the connection between dietary live microbes, non-dietary prebiotics/probiotics, and the prevalence of OA and RA. The graphical abstract of the study is shown in the lower left, and the flowchart participants' selection is depicted in the figure on the right, which excludes minors, individuals with missing variables, and patients with gastrointestinal tumors. The highlights of this study are as follows. First, intake of non-dietary prebiotics/probiotics was positively associated with the risk of developing osteoarthritis. Second, higher intake of live dietary microbes was negatively associated with the risk of rheumatoid arthritis, RA. Third, a mediating effect of neutrophil count and leukocyte count was observed in the association between dietary live microbes intake and RA. The results of this study are as follows. Firstly, there are significant differences in demographic characteristics between the healthy control group and the OA and RA patient groups. Meanwhile, RA patients have a reduced intake of dietary live microbes and non-dietary prebiotic/probiotic, while OA patients have an increased intake of dietary live microbes and non-dietary prebiotic/probiotic. Then, in the association analysis, the results from univariate and multivariate logistic regression analyses indicate that the intake of dietary live microbes is not statistically significantly correlated with the prevalence of OA, but it is negatively correlated with the risk of rheumatoid arthritis and other non-OA joint diseases. Similarly, the analysis results from both univariate and multivariate logistic regression show that the intake of non-dietary prebiotic/probiotic is associated with a significant increase in the prevalence of OA, while there is no statistically significant correlation with the reduction of RA risk. Subsequently, we further conducted mediation analyses. The results showed that neutrophils and leukocytes mediated the modulation of RA by dietary live microbes. In addition, neutrophil number and leukocytes count mediated by 2% and 2.4%, respectively. Further subgroup analysis revealed that the race/ethnicity, alcohol consumption, marital status, BMI, different dietary patterns, and neutrophils and white blood cells may influence the correlation between the intake of dietary live microbes and non-dietary prebiotics/probiotics and the risk of arthritis. In the sensitivity analysis, given the high number of missing values for intake of non-dietary prebiotic/probiotic, accounting for more than 40% of the sample, this study further conducted an analysis excluding this variable, and the relationship between dietary live microbes and RA and other types of arthritis remained consistent with previous findings. To summarize, the study indicated that the risk of RA and other non-OA types of arthritis are negatively correlated with dietary live microbe intake. Conversely, the intake of non-dietary prebiotic/probiotic is positively correlated with the risk of OA. A mediating effect of neutrophil count and leukocyte count was observed in the association between dietary live microbes intake and RA. Race/ethnicity, alcohol consumption, and etc. may influence the correlation between the intake of dietary live microbes and non-dietary prebiotics/probiotics and the risk of arthritis.